single carbon or C1 metabolism focuses on compounds that lack carbon bonds. For example, methanol, methane, or multi-carbon compounds. For example, dimethyl ether or dimethylamine. Okay, these are simple methyl compounds. Although dimethylamine and dimethyl ether has two carbons, there is no carbon bond within the compound. The structure of other examples are shown here. So the focus is on compounds lacking carbon bonds. So we can differentiate the organisms, the microbes that are involved in single carbon metabolism as methylotrophs that utilize C1 compounds as sole energy source for their growth, or methanotrophs that metabolize methane as their only source of carbon and energy. So methanotrophs utilize methane, whereas methylotrophs utilize C1 compounds. So methylotrophs utilizes C1 compounds by oxidizing them to formaldehyde, which in turn can either be used as energy or is assimilated into biomass. Here we can see C1 compounds being oxidized to formaldehyde and then used either as energy or biomass. Good examples of microbes that are methylotrophs are Methanosarcina, which can both utilize and produce methane, Methylococcus capsulatus, which requires methane to survive, and Pichia pastoris, a biotechnologically important model organism that can use methanol as carbon and energy source. Because methanol is more abundant and easily purified and cheaper than sugar carbon sources, methylotrophs are useful in biotechnology for the production of amino acids, vitamins, and etc. The other group of organisms that utilize C1 compounds are methanotrophs, especially those that are able to metabolize methane as their only source of carbon and energy. Methanotrophs occur mostly in soils, mud marshes, oceans, soils, rice paddies, landfills, especially near where methane is produced. Methanotrophs are of interest to researchers studying global warming because methanotrophs can prevent or reduce methane concentrations, as the global warming potential of methane is higher than CO2. There are several subgroups of methanotrophs: Methylococcaceae, Methylocystaceae, and Verrucomicrobiae. First, for methane utilization, there is a reduction of oxygen to water and a concurrent oxidation of methane to a more active species, for example methanol, using oxidoreductase enzymes called methane monooxygenases. These enzymes are soluble in cell cytoplasm or can be as a particulate form in cell membranes. Methane utilization can also be utilized by certain bacteria by running the methanogenesis pathway in reverse. This is especially common in methanogenic bacteria. Other than utilization of methane, methanogenesis is also part of the C1 metabolism. So methanogenic bacteria are only found in the domain Archaea, phylum Euryarchaeota. Examples are Methanobacterium species, Methanobrevibacter species, Methanococcus species. So methanogenesis is a form of anaerobic respiration where the oxygen inhibits growth, in which carbon rather than oxygen is used as an electron acceptor. So you can look at the equation below for the formation of methane. So CO2 works as the terminal electron acceptor. Another example is where acetic acid is used as the terminal electron acceptor to form methane and CO2. Many methanogens live in close association with other bacteria that produces fermentation products such as fatty acids, alcohols, branch chain and aromatic fatty acids, where the amount of carbon is more than 2. A good example would be the rumen habitat, where methanogens thrive. So these bacteria oxidizes them to acetate that methanogens can use. So a phenomenon where one species lives off the products of other species is called syntrophy. So in rumen microbiology, and also in the C1 metabolism, syntrophy or cross-feeding is common. We have methanogens and partner bacteria that perform anaerobic fermentation, where large organic molecules are broken down into hydrogen and acetic acid. Other than rumen microbiome, the guts of termites are also good examples of where syntrophy can exist. So in the guts of termites, we have methanogens and protozoans working together, where the protozoans break down cellulose, releasing hydrogen, which is then used in methanogenesis.